Welcome to Snap Podcast.
Hi everyone, welcome to another edition of the SNEV podcast. My name is Liam Horrigan and today I'll be talking to Charles Lai. He leads a group based in the Institute of Atomic and Molecular Sciences, Academia Sinica in Taiwan. Now, we thought Charles would be an interesting researcher to have on this podcast as, among other things, his group is renowned for developing systems for fluorescent and bioluminescent labeling of EVs. So a general trend we see across the EV field as it's developed is that the development of new technologies and methods for isolating and characterizing EVs is closely tied to our ability to push our knowledge of EV biology forward. Charles and his group recently produced a bioluminescent reporter which has done just that. It shone a light on EV biodistribution in vivo with a level of sensitivity which we would have loved to have 10 years ago. So we're going to talk about these labeling systems and what the future of EV, or more generally EP labeling, could look like. So thanks for tuning into the podcast, and with that little preface out of the way, let's hear from Charles Lai. Thank you, Charles Lai, for agreeing to be talked to today. We've invited you on here to talk about, your technologies you've developed, but it's always kind of fun to hear about how people first got involved in EV research. So do you want to tell us your story there?
Okay, great. Thank you, Liam. And it's a pleasure and honor to be invited to attend this podcast session. So I was very fortunate that I did my postdoctoral training under Dr. Zendra Breakfield's lab. And that's essentially how I was first introduced to extracellular vesicles. Yeah, if we want to shorten the story. And I think when I was under Dr. Breakfield's tutelage, I got really interested in imaging vesicles. Although Dr. Breakfield is a geneticist by training, and then I was very fortunate that she supported my research in exploring how to image vesicles properly. And I think that's sort of how everything started, yeah.
We're often hearing from EV researchers that are based in Europe and the US, but it's good to hear about EV research communities in other parts of the world. So what is EV research like in Taiwan, and what's the community like over there?
I see. I think generally speaking, people are more interested in applying vesicles for therapeutics or diagnosis. At least that's what I see in Taiwan right now. That said, there are other groups of people who are into engineering or engineer vesicles as well. So I think that's what the vesicle communities in Taiwan are doing predominantly. Not so much on the basic research of vesicles, such as the biogenesis or the, for example, the corona effect of vesicles. Not so much in Taiwan just yet, but there may be other groups that I missed.
That's interesting. Is it a very sort of big community, would you say? Like, is it quite interconnected?
I would say it's growing, yeah. So, but again, I think people are very interested in applying them, which is, there are pros and cons. Yeah, it's sort of what the US went through when vesicle first started, I would say back in, 2010. Yeah, I think when people really got excited, they were all jumping into liquid biopsy with vesicles. Yeah, and then so I think Taiwan is kind of going through that phase, but instead of diagnosis per se, I think they are looking more into applying them, especially with regenerative medicine field coming in, that they're really trying to use, say, mesenchymal stem cell derived vesicles for regenerative medicine. But that said, as far as I know, it has not been approved by the FDA in Taiwan yet. Yeah, and then similarly, I think that's the case in the States.
Yeah. Well, thanks for sharing that. So yeah, again, we wanted to invite you on here to talk about the labeling systems you've developed. So for a start, how did you sort of get into that? What was your pathway through research to this point?
Oh, cool. Yeah, so to be honest, when I was first introduced to extracellular vesicles, it was hard for me to be convinced that they actually exist, and it was hard for me to be convinced that they actually confer a biological function. So So to my supervisor's dismay, I told Sandra that I really want to see it. So I think that's what initiated my project. Even though she told me that there were other projects that may be more interesting. Yeah, so I guess to me, like being able to detect and especially in vitro, yeah, I think it's really critical to at least show that they are actually there. And not only that, you can have the spatial as well as the temporal resolution of these nano-sized vesicles. Yeah, so I think that's what really motivated me in the first place. And I think that's what led to the development of the labeling methods. Because when I started my project to visualize vesicles, just like most postdoc, I was going through literatures, and the most predominant method at that time were using lipophilic dyes. Yeah, and then, but funny thing is, like as you go through the manual, yeah, they tell you that they have excellent half-life in vivo, which can last up to hundreds of days. And it makes sense because they were initially used to label immune cells and then to track them in vivo. But that doesn't make sense if vesicles can last that long in vivo. So I think that was one major red flag that came up, is that perhaps we would be tracking the dyes after a certain period of time instead of the vesicles. Right, so I think then I decided to create molecular reporters. Yeah, so when I was under Sandra Brickfield's supervision, I developed a series of fluorescent label for vesicles, and at the same time I also developed bioluminescent-based reporter for vesicle imaging. And through that, we were able to track vesicles both in vitro as well as in vivo. At the with the different series of reporters. Yeah.
So you saw a bit of a gap there. It was the labeling systems that we used before were sort of more general and being applied to the EV field when they weren't specifically made for EVs, and then you thought it was time to develop something more specific.
Yeah, but I think what makes it interesting was that the dyes were really, really helpful in terms of the signal. I mean, the signals are just excellent, but I think it's also kind of tricky, because at one point you kind of ask yourself, is that, wow, are these signals all vesicles? Or are they just like, I don't know, dye residues? And I think that was something, so remember that was like back in 2010, right? So everybody is still trying to find their way to isolate vesicles, and everybody is still trying to identify a more solid method in detecting them. Yeah, so I guess it was a little bit fun, because I was kind of going against the general trend. Because like PKH, for example, it was used in most of the paper around that era. Yeah. And then to kind of go against it was kind of like, But still though, when you look at the signal, you really start asking yourself the question, is like, maybe these are not labeled vesicles.
Yeah,
and of course, over time, the two signal, I mean, the lipids and the dyes may depart. Yeah. So those things kind of came into mind. So yeah, it was a fun process, but also a little bit worrisome, because when you're a postdoc, you're under the pressure to publish good works, and the fact that you're going against the general trend. I was very fortunate that Sandra really provided a strong support with my work at that time. So yeah.
That's good to hear, and glad it worked out so well for you in the end.
Well, we're still working on it. Yeah, we're not saying the dyes, like I always say that dyes are not evil. It's just that you need to know its property and then interpret it correctly. Yeah, so dyes are not bad stuff. It's just that you need to know how to work with them. Yeah.
Yeah. So can you talk us through one of these labeling systems you've developed, like the palm-grep reporter, and maybe give us some insight into the thinking or the processes behind the development?
Oh, of course, yeah. So, as I mentioned before, when I was in Sandra's lab, we developed a series of fluorescent reporter, and then we also developed a series of bioluminescent-based reporter. And when I started my lab in Taiwan, I thought, wouldn't it be nice if you have one single reporter that can do multi-resolution imaging of vesicles from in vivo to in vitro. So I think it's analogy to that is like creating like a Google map. When you use a Google map, you can search the country and then you can go to the city and then you can go to Street View. So that's sort of the idea that I had in mind when I want to create a reporter that can do multi-resolution imaging. So based on that concept, I developed PalmGret, which essentially uses bioluminescence resonance energy transfer. Right. So it's a little bit similar to FRET, but instead of a pair of fluorescent reporters, it's a pair of bioluminescent and fluorescent reporter, such that when the bioluminescent reporter is activated to emit its signal, it can, through bioluminescence resonance energy transfer, to excite the paired fluorescent protein. And under that type of setting, you will have a really good signal-to-noise ratio because you no longer need external excitation to excite your reporter. And this is really critical for in vivo detection of vesicles. Because imagine if you're doing in vivo imaging, typically you would need external excitation light source to penetrate the tissue. So you would get a ton of scattering before you actually hit the target of interest. Then whatever you label within the tissue also need to emit the signal. Then it by itself has to go through all those tissues before you can detect it from the outside. Yet if you skip that process and just inject a substrate to activate the luciferase or the bioluminescent reporter, it by itself will actually emit light. And that light can also excite the paired fluorescent protein. And through that, you get really good signal-to-noise ratio. So we were very fortunate that it worked out that we actually were able to detect very, very minute changes in the biodistribution of injected vesicles. And beyond that, because it has a fluorescent reporter, we can also do orthogonal validation of where the signals are. Yeah.
Yeah.
So that's sort of like a quick gist of the process.
Yeah, no, that was good. Thank you. When I first heard of the PalmGret reporter, it was in the paper where it was used to look at biodistribution in the macaque monkey. I thought that was fascinating.
Yeah, but the one before that was actually published in Advanced Science. Yeah, so the paper that you mentioned was through collaboration with Dr. Ken Whitworth from Johns Hopkins University. So which we were very happy because when Ken approached us about the reporter, I said, yeah, sure, why not? And then interesting enough, like they literally repeated most of the key experiments that we claimed before they try it in the macaque monkey model.
Convenient
replication.
And
I think it was it was really funny because he actually said that, hey, Charles, like, don't mind it that we did this because we were doing macaque monkey model. So we treat every subject as a patient. So we have to make sure that everything is done right and that your reporter is as what you described. And so we were very glad that whatever we reported were repeatable. In their hands.
Because I guess that's one of the major implications with these systems. It's sort of unraveling biodistribution. Sort of where do you see that headed in the near future?
I think it's really important, but it's also one of the more, I wouldn't say the most, but it's one of the more difficult characterization to do for vesicles. I think in a perfect world, it would be nice if we don't have to label the vesicles and then we can track them in vivo.
That would be nice.
Or essentially to track endogenously released vesicles without labeling. And I know that I think this is coming up in the 2024 ISET Tech. I know Dr. Stephen Bopar's group. I think they're creating some non-label based detection of vesicles through second or third harmonic generation and such. But I think in a perfect world that would be what we want, but we're just not there yet. And then to detect them and have the resolution to detect them in vivo is really critical for our understanding in vesicle biology as well as their therapeutic design, because it goes into the pharmacology of vesicles, which is a critical component when you're trying to develop a therapeutic product.
Yeah, that is one of the big barriers to therapeutic application, right? We we don't know enough about biodistribution.
Not yet, at least. Yeah. So, but in terms of biology, I think it's really cool. I mean, for example, under the context of cancer, I think, for example, I think one of the landmark findings from David Leiden's group, they were able to see that, some vesicles travel to certain tissues to educate certain immune cells, for example, or fibroblasts, to kind of pre-educate the pre-metastatic niche, right? And then, but the thing is, and then to this day, like those were injected vesicles. Those were not endogenously released vesicles. So there were limitations there that we can yet replicate in a real world environment. And I think that's something that's really important for us to explore and then for us to understand.
Yeah. I mean, I brought up the macaque paper there, but it must be quite a feeling, developing these systems and sort of seeing them being taken up and applied by other researchers in their own niche research interests. So what is that feeling like, to see something you've made sort of be picked up by other people, and perhaps what is your personal favorite application you've seen thus far?
I would say that it's kind of like a confirmation in a way, such that, like people really find your reporter useful. And in fact, we do share all of our published reporter on atgene.org, which is a nonprofit repository, as you would know. And I think that is one of our, or at least one of my belief in doing research, is that whatever you created can be readily shared among fellow scientists, right? So I think I think having other people's who are able to reproduce your data or your reporter is a really good feeling, to say the least. And I think from there, if they can give us some feedback, we also appreciate it, because that's how we improve. And then, sorry, I forgot your second question.
You would have seen a few different applications of these systems. What was your perhaps?
Right. So I would say it's just what I said. It's really nice that other people can just use our reporter or modify on top of it. I think that's why we make it really open.
Yeah. Have there been a few people who've taken your reporter and just run with it?
There have been. I think they tried. I think maybe, let me think. I think Dr. Edith Buzas from Semmelweis University, I think they have tried our Pong grad to recently report the torn bag mechanism of it's another new way of how small ectosomes are released. So it's a pretty interesting story. I think it was recently published in eLife. And then there were a couple others which are I didn't keep track of them, but other people also took it. But I think not as many people have used them for BioD other than Dr. Ken Whitworth's group. But I think it's something that people can maybe look into, because it's a really powerful tool in our opinion, because as I said, not only did you detect where it goes, you can also do orthogonal validation. And I think that's really important. And I think this is something that the vesicle research group are coming down to, is that you cannot use one single method. To convince at least a reviewer that this is it. You gotta use at least 2 or maybe sometimes 3 or even 4 to say, hey, they all come to the same conclusion. What do you think? And I think our reporter does provide that ability or capability to do so.
Yeah, absolutely right. So what do you see as the next steps in developing fluorescent or bioluminescent reporters, or just, extracellular particle labeling more generally?
I see. Oh, well, you're using the term really precisely. So, so our labeling method is more like is a general labeling method. So it doesn't. So when I was designing the reporter, I was careful not to use, for example, vesicle enriched markers such as the tetraspannins. Reason being is because, again, like imagine back in 2010, everybody is still kind of trying to figure out how the biogenesis or what type of vesicles are. So at that time, I kind of go, why don't I just label the membrane? So it was through that route I decided to label the inner leaflet of the lipid bilayer. And there were 2 reasons behind it. One is that I didn't want to label on the outer membrane, because that might affect the lipid receptor interaction of the nanoparticles. So that was one reason. The other reason is that if we label the inner membrane, they may not be as easily degraded or cleaved off by the proteases or other proteins. Yeah. So through that, we were able to identify the labeling indeed label not just small, they also label large vesicle and also label particles. Yeah. So with that, we decided to continue to use such similar labeling methods, and we're now developing other colored reporter based on bioluminescence resonance energy transfer. And so this was reported in the latest 2024 ISAF in Melbourne. Yeah, and then we are now trying to understand that what would happen if you have multiple vesicle populations. And this is actually how vesicles are released endogenously, right? Like, I don't think the cells will only release one size of vesicles at once. So supposedly the cells, or especially for our research under the context of cancer, for example, I believe that cancer cells will release any type of vesicle to its own advantage. And then, so in fact, I think our latest study, which we published, is that we found that triple negative breast cancer, which is a form of very aggressive breast cancer cells, they actually release more, we call it big vesicle at that time, or you can use large vesicle. We actually found that those triple negative breast cancer cells actually release more bigger vesicle when compared to the benign epithelial counterparts. And then, so from there, we actually got the insight that, so if we know that certain cancer cells release other vesicle populations, then wouldn't it make more sense if we can track them simultaneously? We can track the small and the big. And then could they potentially interact with one another? And if they do, how does that affect the function that they confer through vesicle? Yeah, so I think that's something that we're trying to explore right now, is that yes, we know that there are small vesicles, we know that there are larger vesicles, and I think that's not only being reported by us, also by others. But if you detect them simultaneously, do they have interactions? And is it 1 + 1 = 2? Or do they have other dynamics that we are not aware of? And that's something that I think can hopefully be elucidated by using more than one color. And indeed, we now have 2 colors that can do multi-resolution imaging of the vesicle population.
And how would you be labeling these 2 populations separately using these 2 different colors?
So this is a limitation. We still have to generate a stable line. So we have to go back from to the donor cells. We have the donor cells expressing 2 separate reporters, right? And then we isolate vesicles from these cells for the subsequent tracking. Yeah. So yeah, there's always a limitation that we have to overcome. I think another, so this is sort of like extension of one of your questions, is that, yeah, it would be nice if we can label vesicles that's already being isolated. And I think that's where the dyes will come in.
Oh, yeah.
Yeah, or I think other people are also trying to develop other methods in labeling vesicles through that route. But it's not that easy yet. Yeah. But there are ones such as, I think a more accurate one perhaps is the CFSE, the one that would the dye would be permeable and go into the cytosol of the vesicle, and then the enzyme will cleave up the tail for it, the esterase will cleave up the tail for it to remain inside the vesicle. I think that method might be a little bit more advantageous than other lipophilic dyes in terms of the specificity of the labeling. Yeah, and I think there are other papers that were trying to do comparison on it, but we didn't use them per se in our hands. So yeah, so I think, but that's something that our method cannot reach at this current moment. Yeah.
I mean, aside from just different colors, the bioluminescent labels, sort of what other future directions can you see with those in the next 5-10 years? Are there various other things about them that can be tweaked and improved?
I'm sure there are. Just that,
yeah.
I mean, so I think we have spent the past almost 3 years on multiplexing the bread reporter, which have proven to us to be very, very challenging. Yeah, it's sort of like once you have two reporters, the range of difficulty is like exponential.
Yeah.
Yeah. When it's only a single reporter, it was relatively straightforward. But once you have two, then you kind of go, yeah, like you have to avoid signal crosstalk and substrate crosstalk and such. So that took us a while. I think in the future, yeah, I mean, like there are many great scientists who continue to develop bioluminescent reporter as well as red reporters. So with new ones coming out, and given that they don't exhibit signal or substrate crosstalk, then I think we there is a potential that we can generate more vesicle reporter to add to the palette. But it's not easy because most reporters, at least currently, are sort of created through evolution. Yeah, so it's not like they created a de novo bioluminescent protein out of a sudden. Usually they like create and modify. It's sort of like the fluorescent protein reporters. Yeah, like they evolve through the different generations. And then because of that, a lot of them share substrate crosstalk. So I think that's one of the major challenges. Yeah, but I do know that there are many great scientists who are working on those. I think, but just to use the existing methods that we have right now in exploring the complexity of vesicles would be key, because I think I think many great scientists have done a lot of great work on vesicle in vitro. And I think to overcome the technical or imaging barrier to go in vivo is something to look forward to. And but there is a challenge between preclinical and clinical research. Yeah, I think to image vesicles in patients would be phenomenal. So as I mentioned before, Dr. Stephen Bopar, perhaps their tools can do that. I'm not sure yet. Yeah, I think that's something that people can look forward to. I think they did have a couple paper reporting on that, at least in their preclinical models. Yeah, right. But preclinically, I think not only with our reporters, but I think other people's reporters will also come into play to explore the vesicle dynamics in vivo more and more. I think that will be something that the field can invest more on. Yeah.
What are the other limitations and barriers for live imaging and tracking? Microscopy and whatnot. What are the interplaying factors here?
I see. Like let's see. I would say specificity in labeling is key. Right. I think everybody knows that, and I think just by coincidence, like as we were talking about the dyes, it was later when the flow cytometry experts start to see there were dye aggregates from the dyes. I think so. Specificity of the labeling is key. The quantum yield is also key. Because we are trying to study nano-size entities. So if your labeling reporter or dyes are not good enough, usually it's really hard to have a really good signal-to-noise ratio. Yeah, so I think those are the things that people need to think about when they're designing their experiments. And because that would directly relate to the model or your experimental model. If you're doing in vitro, usually it's much easier because you can just use your confocal or other microscopy to image it. But if you're trying to image them in vivo, it's a whole different ballpark that you have to think about. Yeah. So I don't know if that answered your question.
It's close enough. Yeah, so I mean, we talked a bit about biodistribution, but what are the other sort of research questions that you can see, new developments in this area kind of making a bigger impact?
Yeah, I think we were on this a little bit before. Is the endogenous vesicle? Yeah, I think it still is the biggest conundrum for us to study. Yeah, because put it this way. As a disease is being formed, let's say like under the context of cancer again, detection of cancer cells at early stages are really difficult. So conceivably, detecting vesicles released from cancers at early stages is even more difficult, right? Because the signal are just so minute, right? So like if you want to study how vesicles play a role during a biological function from the beginning to, say, the terminal stage or to the effective stage, it's really hard to track that until the phenotype is really, really prevalent. And I think that's something that we want to overcome, but it's still pretty challenging in my opinion.
What about around vesicle fusion? Are these reporters really making, or future reporters making much of an impact there?
You mean in vivo?
Yeah, and in vitro.
I think in vitro will be a bit relatively easier. I'm not saying that's easy, but relatively. But I think in vivo will be tough, at least for now. Yeah. Yeah, I mean, there were other scientists who used like contrast agent to label vesicles, but those were largely labeling based on isolated vesicles. Those were not endogenously released vesicle tracking. Yeah. So I still find it pretty challenging to study how naturally or endogenously released vesicles work in the body.
Yeah.
Yeah, and I think it's a very exciting field. Yeah.
A lot. Yeah, I know. There's fruits ripe for picking there. Many questions to be answered. So my last question for you. I mean, we've kind of already covered this a little bit, but the latest MySeb guidelines, they draw the distinction between EVs and NVEPs. And the Palmitoyl label, it is, a pan-EV label. I mean, I've also seen that it's been purportedly used to label eczemias, an NVEP. But I was wondering if you might see any more of these sort of, somewhat ubiquitous markers like Palmitoyl, but they are perhaps, trafficked into different EP populations. Beyond obviously just using, fusing a label to some membrane protein like CD63 or something like that,
So your question is that are there other labeling moieties?
Yeah, sort of in the near future.
Okay. I would say other people have also tried other membrane labeling method or other methods. I think palmitoylation is one of the, if I'm not mistaken, one of the two main acylation process, right? I think there were other people who use a combination of palmitoylation as well as myristoylation. It was a very conventional way of labeling membrane proteins back in the days. Yeah, it was during the development at that time that I thought the sequence was too long, so I just cut off the myristoylation and I tried the palmitoylation. And then I actually did a side-by-side comparison between the palm and the myristoylation tag for some protein. And in fact, like the signal literally co-localized by about 90%. And then so I thought, well, I didn't want to have such a long tagging sequence. So I chose the palm, and I think just by a stroke of luck that later reports show that, hey, palmitoylation was actually one of the common moiety in the vesicles. I have not seen so much about myristoylation, but I do know that people have tried that, or a combination of the two. Yeah, so I think that's one way in labeling. I don't recall there's a paper that compare which one is better than the other yet, but I chose palm at that time also because it's a reversible process, whereas myristoylation is not reversible. Yeah, so I think at that time I was trying to make sure that if the palm label gets degraded, it would be easily detached. This is just conceptual thinking. There is no
that's what we're all about here. It's a good place to speculate.
Yeah. But myristoylation at that time, I was worried that the dot or the reporter gets recycled over and over again and would not dissociate from the membrane. And therefore, it might actually screw up the spatial temporal resolution. Yeah, so at that time, I didn't choose myristoylation to go on. But to this day, I don't have hardcore data to discern which is better than the other. Yeah, so, but yeah, I think like other scientists are also developing ways to label vesicles better. I'm not saying that our way is the best. I think it comes down to what method and reporter will work best for your experiment and would better address the question you're trying to elucidate. I think that's what it comes down to. Yeah, there's no one best solution for vesicle research. And I think that's a conclusion that we come to over the years.
All the time, with every method.
No, it's just not there yet.
Yeah. But anyway, this has been very enlightening. Thank you. It's a fun little area to shine a light on.
Yeah, and I think that's it's an important field. I just think that not many people want to traverse this path because it can get quite complicated. Yeah. Yeah, it's not just 2D imaging. It's a lot of imagining how you gotta have an idea about how microscopic anatomy works, and then you gotta kind of appreciate that this is an organism. So and then just imagine that how these vesicles may be trafficked to yield a biological function. I think that by itself just gives me a lot of excitement.
Yeah, definitely.
And then because this also goes into dosage. It goes also into the duration of the vesicles, right? And I think that's something that you can easily manipulate in an in vitro condition, but what's going on in vivo, you don't know. Yeah, or at least we don't know. The best we can do right now is to give, say, for example, IV or IP or IM injection to kind of, and hopefully mimic the physiological condition, but we still don't really know their function or their property under a more physiological condition for vesicles.
You're exactly right. Yeah, these are questions that will need to be answered before we can really see some therapeutic rollout en masse.
Perhaps, but that's what we're here for.
Yeah,
right?
Absolutely. Well, thank you again, Charles.
Thank you very much, Liam.
And thank our audience too.
Okay, thank you. I hope it interests them.
I'm sure it will.